Okay, so my name is Wendy Suzuki, and I just want to say that it's both a real pleasure and an honor to be here to help celebrate all of the wonderful contributions of James Ronk to this field that we all love. And I'm definitely of the camp of providing kind of cool things that the hippocampus does beyond place and kind of head direction. And I want to focus on very new data. I've never talked about this before, about how the hippocampus, and here I'm going to be talking about non-human primate hippocampus, contributes to learning from errors. So if you PubMed or if you Google error-based learning, what you'll learn is that we've studied this a lot in humans. Learning from errors is a really, really good way to learn, and you'll find lots of studies on the role of the medial prefrontal cortex and the anterior cingulate cortex on detecting these errors that contribute to this ability to learn based on errors. You'll also read a lot about the role of the striatum in error-based learning. But it's learning. And so you might ask, well, maybe the hippocampus plays a role. And we can go back to this classic paper that so many people have gone back to find evidence that back in 1973, Jim had noticed things that predict these later findings and the findings that I'm going to be presenting today. And in particular, I'll highlight the approach consummate mismatch cells that have this idea of an error detection. What's going on there? Well, later, the idea of the role of the hippocampus in error-based learning was made more formal in the form of computational models, first by Lorenz and Bujacki, and then more recently by Ketz and O'Reilly. And particularly the Lorenz and Bujacki paper is really useful because it has very specific predictions about what they thought these areas might be doing during error-based learning, although there wasn't a lot of physiological evidence for it. So what are the predictions? The predictions were, one, the entorhinal cortex was the key area for detecting errors. Two, entorhinal cortex induces synaptic modification in the hippocampus. And then three, the hippocampus, once this learning from errors happens, the hippocampus then entrains long-term memory signals in the entorhinal cortex. Well, my postdoc, Shipi Ku, got very interested in these specific predictions as she was doing a kind of side analysis project on her data and some of the previous data gathered in the lab by Dr. Eric Hargraves and Dr. Sylvia Worth. And she was analyzing kind of the classic experiment that we've done in the lab for a long time, the first kind of task that we focused the lab on. And this is an associative new learning task. We call it a location scene association task. And in this task, animals first, these are monkeys again, first fixate a central fixation spot right here. Then we show them a complex image, and superimposed on that image right here are 4 targets. You can see north, south, east, and west. They have to maintain fixation, and then the picture disappears. The targets remain on the screen for 700 milliseconds, and then the cue for the animal to move his eyes and choose one of the targets is the disappearance of that fixation spot right there. And when that disappears, that's his cue to move to a particular target. He doesn't know which target is correct. So this is a trial and error learning situation dependent on the hippocampus and medial temporal lobe. And we know a lot about what cells in the hippocampus and the entorhinal cortex is doing. This is to remind you we're now moving to non-human primate. This is a ventral view of primate brain, where you can see in white is the location of the hippocampus just deep to the parahippocampal cortex there. And all the data that I'll be telling you about are from the hippocampus here, as well as from the entorhinal cortex. Well, we also know, because we've been studying this task for such a long time, we know a lot about the behavior of this task. So this is an example of a behavioral learning curve of an animal learning one brand new scene. Let's say it's that gorilla scene that we looked at. On the x-axis are the number of trials that they got, that they completed. The red dots up here indicate whether that trial was completed incorrectly in red or correctly in blue. And on the y-axis is probability correct. And the red line shows shows a dynamic logistic regression algorithm estimating the trial-by-trial percent correct. So we know a lot about how the animals are learning, and we've studied this a lot, but we realized we never asked the question, are these animals learning using an error-based learning strategy? So that's exactly what Sheepy did. And it's very simple to do. You can do the same analysis you do in people. That is, you take error trials and you ask, on the trial after an error trial, what was the behavioral performance? And you concatenate all those and you compare that to the behavioral performance after a correct trial. And all of this analysis was done basically in the first half of the learning curve when most of the error trials are happening. And so she asked, based on this analysis, very standard analysis, is there error-based learning in the location scene task? And so when she concatenated all those percent corrects after a correct performance, she got about 70%. Then she did it for the errors. And she found significantly improved performance. So animals are performing better right after an error compared to right after a correct, which led us to conclude that we actually have a wonderful example of error-based learning. And not only that, we have neural activity in both the hippocampus and the entorhinal cortex, which allows us to address these major predictions in a physiological preparation. So the first question, do we see error detection in the entorhinal cortex? And actually we had evidence for how to look at this from previous studies in the lab. And that was on a separate, a different association learning task, an object-place association learning task, where they have to learn object-place combinations. And here we started looking at what's happening in the reward and ITI period of the task. To see whether these cells actually would signal, told us something about the outcome, whether the trial was completed correctly or incorrectly. And we were stunned to find that in this study focused on the hippocampus, 50% of all the recorded hippocampal cells signaled trial outcome, that is, whether the trial was correct or wrong. And we found two flavors of these trial outcome cells. This is all published, a study by Sylvia Worth. Half the cells increase their activity in that intertrial interval following a correct trial relative to error trials. That's the one on the left. And on the right, you see cells that signal error detection. They increase their activity on error trials relative to correct trials. But this was a different task. We had never asked whether there was this kind of outcome signal in the location scene task, and now we had a framework from the model predictions. So that's exactly what Sheepy did. And she started with the entorhinal cortex because that's where you should see strong error signals. And what she found is 45% of all the recorded cells in the entorhinal cortex signaled outcome. Of those, the majority of them, 67%, signaled errors. Increase in activity on errors relative to correct. Again, this is in the intertrial interval after the animal knows whether it got it correct or wrong. And the other, the remaining 22% signaled correct trials. In the hippocampus, we also found error detection and correct detection, but you can see the differential responses are much weaker, and there are many fewer cells in the hippocampus in this task that are signaling outcome. So let me just jump to here and say, for the first prediction, is there evidence in a task where monkeys are using error-based learning that the entorhinal cortex detects errors? Absolutely, very strong error detection. Let's move on to the second prediction. That's not just for error detection, but what about synaptic modification? What about learning signals that this model says should be happening in the hippocampus? So again, we'll go back to the learning curve, and I showed you behavioral evidence that after an error trial, those red dots, the animal is performing significantly better right after an error relative to right after a correct trial. And we asked whether that also corresponded to a change, a shift in the response properties of hippocampal or entorhinal neurons. What is a learning signal that we might want to look at? We wanted to look at the selectivity of these neurons. That is, the animals have to discriminate between all the different scenes that they're learning. I don't think I said this. Animals are learning multiple scenes concurrently. So one way that the hippocampus could signal a shift that would help them perform the task is if these neurons became more sensitive to the differential they fired more differentially to the different scenes being shown. So we looked at a selectivity index following an error trial compared to following correct trials. Again, all early on in learning. So what I'm going to be showing you for the hippocampus is a plot comparing the selectivity index after errors compared to the selectivity index after correct. And what we see, focus here, this is happening during the scene period, is a shift. After error trials, the selectivity index, shown right here, is significantly higher in these hippocampal cells compared to right after a correct trial. This is the significance right here. It's only significant during the scene period, not during the delay period. So we have evidence of early hippocampal representation, plasticity, during this error-based learning. Does it happen in the entorhinal cortex? No. We found no evidence of changes in selectivity in the entorhinal cortex using the same exact measure. So now we have evidence for early learning, early changes in the hippocampus in this error-based learning task. Then we want to ask, how selective is this? And we realized we had another task that had many of the same components that we had never asked, is this task being used, being learned in an error-based learning way? So Shipi did the same analysis on the object-place association task. And before I tell you what the answer was, I have to say that this task was significantly more difficult for the animals to learn. It took them significantly longer. They made significantly more errors. But theoretically, it was still associative learning task, and when she did the behavioral analysis, what she showed is that this task was not an error-based learning task, it was a correct-based learning task. They learned more after a correct trial in this task than after an error trial. So now we can turn to what is the prediction for any neural activity? And here's what we wanted to find. Because this is a correct-based learning task, we did not want to see any increase in selectivity after an error trial on this task. We might expect an increase in selectivity after a correct trial, but not after an error trial. And what we ended up finding when she did this next analysis, we kind of took bets on what we would find, was no change in the selectivity index in this task in the hippocampus after either correct or error. So there is specificity in the sense that for our error learning task, we only for the error learning task do we see increases in selectivity after error trials, but not for a correct-based learning task. So we can answer the second of these three predictions. The prediction was entorhinal induces synaptic modification. Of course, we didn't look at that directly, but we looked at the synaptic modification in the hippocampus, and the answer is yes. We found evidence in an error-based learning task, and we found specificity for that error-based learning task. And finally, the third prediction. Can we find evidence that the long-term memory signal might be conveyed by the entorhinal cortex? And here we had a lot of experience looking at long-term memory signals in these kinds of tasks. That was basically what my lab has been doing for the last 18 years. And so what did we look for? So this is that same learning curve for the location scene task, and I can show you one of our classic findings, that in an individual hippocampal cell during this part of the learning curve, clearly the animal has learned nothing here. And if we superimpose neural activity in the hippocampus relative to baseline, which is the white straight line, you see that there's no change in neural activity during these trials where behaviorally shows that the animal is not learning anything. But clearly, there's a huge behavioral change at this point of the trial, and we can see that, that there's huge change in neural activity in parallel with learning. We called these cells changing cells, and we know that this represents a shifting of selectivity. These neurons at the end of the trial here, when clear learning has been shown, are much more highly selective relative to the beginning of learning. So are there changing cells in this new population of cells that we recorded on the same task, just a kind of internal check? And the answer was yes. In the population of hippocampal cells, including new hippocampal cells that Shipi had recorded, we found almost identical proportions of changing cells. In the entorhinal cortex that we hadn't looked at before, much smaller, just above chance significance of changing cells. They were there, but clearly more changing cells in the hippocampus. But that wasn't the final answer for the entorhinal cortex. We had another clue for how entorhinal cells might be signaling long-term memory. Again, at the end of the trial, when learning is really high, what do entorhinal cells signal? And this clue comes from going back to this object-place association task. I told you that there's lots of outcome cells. They're signaling correct or error cells, error outcomes. And what we asked is, this is a differentiation during the intertrial interval period, and we asked, well, what happens on the next trial for these error up cells, for example? Do they become more selective on the following trial during the task period itself? And again, this is published data. What we found for this task, remember it's a correct-based learning task, we found that it's the correct up cells during the task itself that change their selectivity with learning. Another learning effect here. The error up cells don't, control cells don't, and if you don't get learning during the session, you get no change in selectivity. But that was the object place task. Let's go back to our location scene association task. And what we find is in the entorhinal cortex, it is the error up cells that are changing their selectivity so that they are more selective at the end of the learning session than at the beginning of the learning session. So another kind of long-term memory signal. So, and we, of course, looked at the hippocampus as well, and hippocampal cells. Remember, they didn't have a lot of outcome signals to begin with. They didn't change with learning. So now we can address the third prediction. Does hippocampus entrain long-term memory signals in the entorhinal cortex? Well, what we found was two different long-term memory signals, one in the hippocampus, those changing cells, and one in the entorhinal cortex, those changing error up cells. So I find these findings exciting for a number of different reasons. One is error-based learning I think it really helps to bring the medial temporal lobe, both the entorhinal cortex and the hippocampus, into the error-based learning field, in particular, the important role of the entorhinal cortex in error detection, which kind of the main players these days are stay up in the prefrontal cortex, medial prefrontal cortex, and anterior cingulate cortex. I think that this is a really exciting time to be looking at it because error-based learning is one category of learning from feedback, and there's a lot of exciting work being done by people like Daphna Shohami and Mike Shadlin talking about interesting and important interactions between the prefrontal cortex and parietal cortex and the hippocampus in a kind of learning from feedback, which other people have called decision-making. The other thing that you might think about in this very strong error detection in the entorhinal cortex is reinforcement learning that is based in the striatum, of course. And ironically, this whole set of data that I just described for you from Shipi Ku was a side analysis project from her major project, which has been to record individually and simultaneously in the caudate nucleus, shown right here in the human brain, and the hippocampus to look for reinforcement learning signals in the medial temporal lobe. And in fact, I'm not going to talk about that, but she has exciting new data that there are many more reinforcement learning signals in the hippocampus than previously indicated, including those that occur earlier in time than those in the striatum, at least in our hands. So I think this really helps to bring, I was so excited to see all of the exciting findings on imagination in the hippocampus that Neil Burgess talked about the beautiful auditory 1D environment responses in the hippocampus that David talked about, and of course the time cells that Howard talked about, and the motivation signals that Matthew talked about. And I think it's a really exciting time for the hippocampus, expanding out beyond to these really, really interesting things, expanding out to cognition. And we can look back to Jim's beautiful studies to see some really smart early observations of that. So thank you very much for your attention.